Actually designed.
Welcome everybody. So I'd like to welcome everyone to the Biomedical Research Institute Maurice Strauss Distinguished Public Lecture. And we're very privileged to be able to bring this program to the scientific community, the healthcare community, and all the guests from Greater Roanoke, due to the generosity and lasting legacy, of course, of Mr. Maurice Strauss. Before I introduce today's speaker and explain tonight's presentation, I want to take a minute to put in a plug for the next speaker in the series. That'll be in January, on January 23rd. We've learned to avoid December because of the holiday crush and so forth, so we'll jump from November to January. We'll have Dr. Regina Barzilay from the MIT Computer Science and Artificial Intelligence Lab, and she will present Rethinking Human Health with Artificial Intelligence, Solving Old Mysteries with Modern Tools. I'm sure it'll be a very exciting talk, and we'll send out announcements. Hope many of you be there. So it's my pleasure to introduce tonight's speaker. Notice the speaker isn't standing here. I see the speaker is on the screen. I promise you this was not a bait and switch, something that was intended. The speaker was trying to get here from New York and had to change planes in Washington, D.C. at Dulles Airport. And guess what? It didn't happen. The plane was gone when he went from gate A to gate B. He so graciously agreed, however, to sit in a car for hours from Dulles Airport to go to our Children's National Research and Innovation Campus site, where he's sitting right now, and to sit there and to present his talk to us live, semi-live. And then he will eat a slice of pizza instead of a wonderful dinner he would have. And then he will get back in a car and spend 2 hours going to Dulles Airport, and tonight fly back to New York. So I can't tell you how much we appreciate our speaker. Sorry, this happened, things happen beyond our control, but I'm sure it'll still be a wonderful, wonderful evening. So let me now introduce this evening's speaker, Dr. Luis Diaz Jr. Dr. Diaz is the head of the Division of Solid Tumor Oncology and the Greer Family Chair in Medicine at Memorial Sloan Kettering Cancer Center, and he's professor of medicine as well at Weill Cornell Medical College. Dr. Diaz studied microbiology at the University of Michigan, where he also did his medical training. He then completed his residency in internal medicine, a fellowship in medical oncology, and subsequent research fellowship at Johns Hopkins, where he worked in the lab of Bert Vogelstein. For those of you who know that name, you know he's a giant in the field. Dr. Vogelstein is one of the great figures in modern molecular medicine, who was the first scientist to elucidate the molecular basis of a common human condition, showing that colorectal tumors result from gradual accumulation, of genetic alterations in specific oncogenes and tumor suppressor genes. Anyway, Dr. Diaz has been recognized by his peers for his many contributions with a number of awards. I'll just name a couple. Received the Pioneer Award of the Precision Medicine World Conference, the American Cancer Society Heroes of the Year Award, the Giants of Cancer Care for Translational Medicine Award, and the American Association of Cancer Research Award for Outstanding Achievements in Translational Research. He's an elected member of the National Academy of Medicine, the American Association for Cancer Research Academy, and the American Society for Clinical Investigation. He was appointed by President Biden to the National Cancer Advisory Board of the National Cancer Institute and he served as director of the American Association for Cancer Research Board of Directors. Dr. Diaz led landmark studies, including the study of PD-1 blockade and microsatellite instability in colorectal cancer, led the first successful trial of immunotherapy in colorectal cancer, which has led to the drug pembrolizumab receiving FDA breakthrough designation in treating refractory colorectal cancer with mismatch repair deficiency. He's also led in the use of circulating tumor DNA for early detection of tumor recurrence and monitoring emergence of therapeutic resistance in the blood. Before I turn it over to Dr. Diaz, I just want to share with you the following. Our director of the Fralin Biomedical Research Institute Cancer Research Center in Washington, D.C., where Dr. Diaz is sitting at the moment, is Dr. Chris Horrigan, who many of you know. Chris, unfortunately, is traveling out of the country at the moment. It isn't there, ironically, with Dr. Diaz being there. But Chris shared with me the following. He said, I've known Luis for over 17 years when we were both docs at Johns Hopkins. I can confidently say he is among the most truly innovative cancer physician scientists I have ever met in my entire career. He has taken his ideas all the way from the lab to the clinic to first-in-class historic and generalizable FDA approval. This is someone who has really delivered. So without further ado, please join me in welcoming Dr. Luis Diaz.
Thank you very much for that kind introduction, and thanks for everyone being patient with me. Even though I'm stuck here in DC, I'm really excited to be here and to be able to share some of our work with you. Can everyone hear me okay?
Yeah, we hear you fine.
Great. So I'll be talking about novel clinical applications of cancer genetics, but this has really been something that's evolved, I'd say, at least over my career for 20 plus years, but it's something that began well before me. And a lot of the roots of this work were started in the basic science laboratories, not just of my mentor, Bert Vogelstein, but many scientists all around the world for many decades before even this began. So I'll get started. These are my financial disclosures, primarily around the use of immunotherapy with certain genetic signatures, as well as with circulating tumor DNA. I'm also on the board of directors of Quest Diagnostics. So let's begin with just a very simple slide about how does cancer evolve. And our understanding of cancer evolution is that you transform a normal state into a pre-malignant state to a localized invasive cancer, then that eventually becomes metastatic. And if you look at these different states under the microscope, you see really classic changes histopathologically under the microscope. And then if you look at the DNA that underpins these changes, what we see is an association with mutations that are either oncogenes, so activating or making the cell grow, or inactivating mutations that turn off the brakes on cells. So mutations that cause cellular acceleration, or mutations that stop the cell from dying when it should die. And these mutations accumulate in key genes and key locations that either turn genes off or on or off. And this really happens over the course of decades. So when a patient comes to my clinic and says, Doctor, how long has this been in my body? The traditional solid tumor, we believe, has been there for 10 to 15, maybe even 20 years or longer. And this has implications for things like early detection and screening. And so this evolution really has taken time to develop over time, and these mutations have really taken hold over time. So over the past, I would say, 30 to 40 years, we've really began to define where these mutations occur. And we call them somatic mutations because they exist in precancer cells or in the cancer cells, but they're absent in the normal cells. And that's how we define which mutations are really cancer associated versus those that are hereditary and pass on from generation to generation. And over the past 20 years, there's been an evolution in what we understand about these cancer mutations. And I show this because virtually every tumor type in men and women and adults and children, and actually even in non-humans, has been sequenced. And we understand the landscape or the mutational landscape across these different tumor types. And we found these associated mutations are present in these tumor types. And we think that that is intrinsic to really the etiology of these tumors. So what do we do with all these cancer mutation information? There are over 2.5 million different mutations characterized at this point. And they're cataloged now across the different tumor types. And what was really the goal in doing that, and what was the understanding that we've developed? I'm going to be talking from a therapeutic and a diagnostic angle, what we've tried to use this information for. And I would say that the dream was that we'd find these mutations and that we'd be able to develop a very precise therapy for these mutations. We'd give that therapy to a patient whose cancer had that mutation, and the tumor would melt away. Of these 2 million plus mutations, there's probably a dozen drugs that have successfully done that, and only in rare cases have they been curative. Typically, they've extended life, but they haven't been the revolution that we thought would be. The second way that we can utilize these mutations is as prognostic markers. And so that's to tell a patient or inform a physician or to inform a research cohort that a tumor is more aggressive or less aggressive than its counterparts. And that can be helpful in not only therapeutic decision making, but also important for the patient to know, this cancer is not one that's going to take you away in the next 2 to 3 years. It's one that we can wait on and we don't need to do any therapy on, or hey, this looks like a very aggressive cancer. Let's treat it more aggressively. What I'm going to talk about is more novel applications that really have emerged over the past, I'd say, 10 to 15 years. And the bulk of it is going to be using these mutations as immune antigens. And I'm going to touch upon briefly about using these mutations in the liquid biopsy area. And I'd say this story really begins with using mutations as immune antigens in 2010, 2011, 2012. That's when we began to see that immunotherapy was really having an effect and that it wasn't just a transient benefit, that it was here to stay. And we were seeing drugs that targeted these molecules on the surface of immune cells called immune checkpoints that put the brakes on the immune system, that if you could block those receptors, you could activate a potent anti-tumor immune response. First in melanoma, then in kidney cancer. And at first we were like, oh, we knew melanoma was sensitive to immune stimulation. With agents like cytokines like IL-2, they would respond. And then in kidney cancer too, we saw responses and we were like, oh, that's an immunogenic tumor. We knew they would respond. But what really caught everyone's attention was when these agents began to work in lung cancer. We had never seen lung cancer have any sort of immunophenotype that would be responsive to immunotherapy. And we were seeing these immune checkpoints working in lung cancer. And what we knew at that time about mutations and these different tumor types was, well, very simply, that some tumors had a lot of mutations and other tumors had fewer mutations. Liquid tumors like leukemias, relatively few mutations. Pediatric tumors, relatively few mutations. Sporadic adult tumors like colon cancer, like breast cancer, like gastric cancer, pancreatic cancer, had about 75 to 100 mutations for what we call the coding exome, what's expressed in the genome. And then mutagen-induced cancers like melanoma and lung cancer had many more mutations, between 150 to 300 mutations for coding exome. And so the initial thought was that, oh, these tumors like melanoma and lung cancer are responding maybe because they have more mutations. That hadn't been proven at this point, but that was just a thought actually that our group had as well. And we were very interested in these tumors that were mismatch pair deficient because they didn't have a few hundred mutations. They had thousands of mutations per cancer genome. And if lung cancers and melanoma were responding well, could those respond even better? And so a little bit about those mismatch pair deficient tumors. So it comes in two forms. There's a hereditary form called Lynch syndrome, and there's a sporadic form called just sporadic mismatch pair deficiency. In the hereditary form, you have a patient who has one of the alleles of mismatch repair protein lost, and they acquire a different hit to inactivate mismatch repair. In the sporadic form, you lose both alleles or both sides of the gene alleles that inactivate mismatch repair. And it's more common in a hereditary, I mean, in the sporadic form, but the hereditary form is also common, but only about 10-15% of cases. And what happens when you inactivate mismatch repair is that during DNA replication, you can't recognize and repair spontaneous mutations. And this results in the accumulation of mutations that are single base pair mismatches or short insertions and deletions. And this accumulation leads to thousands of mutations being harbored in these cells. And they're enriched in these repetitive elements called microsatellites, and you get this thing called microsatellite instability. And people search for microsatellite instability as a marker for having mismatch repair deficiency, and you get an exceptionally high mutational burden. And these clues were first evident really in the 1990s or even before that, when multiple groups identified germline mutations associated with Lynch syndrome. And this was work done by my mentor, Bert Vogelstein and Albert Del Chapelle and Laurie Altman. And what they found was that these familial cancers harbored alterations in these short repetitive elements, suggesting multiple alterations throughout the genome. And these families had these markers. And when we looked at these families, or when they looked at these families, they found that these families who had hereditary forms of colon cancer had replication error defects that were found in these microsatellites, whereas ones that didn't, colon cancers that didn't, almost had no alterations in these microsatellites. And Nick Papadopoulos, who I worked with as well, found one of these genes associated with Lynch syndrome. And that allowed investigators like Ramon Parsons and Bert Vogelstein, Paul Modrich to show that these cells that had these replication error defects with these hereditary mutations had a high hypermutability profile. Also in the early 1990s, Stan Hamilton, who was a pathologist at Hopkins, described or really compiled what pathologists had known for decades, which is that patients with familial cancers, particularly colon cancer and endometrial cancer, if they saw a dense lymphocytic infiltrate, immune cells infiltrating the tumor, they predicted that these were the tumors that were hereditary, and they likely had these replication error defects. So pathologists knew even ahead of the molecular biology that something was going on with these hereditary mutations. So the hypothesis was that if these tumors harbored thousands of mutations, and if these mutations were transcribed and translated into proteins, they'd be looked as foreign antigens. And we knew that these mismatch repair deficient tumors were highly inflamed. So what if we married this immunotherapy breakthrough with this genetic observation, and could we unlock a very potent anti-tumor response? And so that brings us to the first area, which is, could we treat tumors with this approach? So with Young Lee at Johns Hopkins, we designed this very simple clinical trial. We'd take colorectal cancers that were mismatch repair deficient. These are metastatic patients, and compare them to colon cancers that were mismatch repair proficient. The difference being the mismatch repair deficient tumors had thousands of mutations, and the proficient tumors had few, less than 100 mutations. And then we knew that this signature also occurred in non-colon cancers. So we had this other bucket of mismatch repair deficient tumors, but again, with expected to have thousands of mutations to see if they'd respond. So we initiated the study in 2013 and completed enrollment in 2015. We used a drug called pembrolizumab, which was an anti-PD-1 drug. This is before it was approved for any cancers. And we used a relatively high dose, 10 milligrams per kilogram every 2 weeks. And we treated patients who failed all standards of therapies and the evidence of disease progression. So while we were running this study, Tim Chan and Alex Slider at Memorial Sloan Kettering showed that in melanoma, patients who had more mutations did better than patients with fewer mutations. And Tim Chan also showed that in lung cancer treated with immune checkpoint blockade, those with high mutations did better than tumors with low mutations. That was a retrospective study. They were trying to understand what was going on. And Drew Portal and Nick Losa at Hopkins showed that these mismatch repair deficient tumors had elevations of the immune checkpoints. So everything was in place for this clinical trial, and we were really excited to see the results as this paper came out. And what we found, and was remarkable, is that these responses were very fast. And this is a tumor marker called CEA that you can measure in the blood. And in red were the patients with low mutational burden. The tumor markers went up very quickly, meaning that these were the low mutational burden, that tumors were growing really fast with the low mutational burden patients. Whereas the patients that were mismatch repair deficient with a high mutational burden, we saw the tumor marker normalize, meaning that the tumors were shrinking, at least what we saw in blood. And it was very quickly. In terms of the radiographic response, those are proficient, low number of mutations. The tumors grew from baseline in virtually all the cases, except a few. And the deficient tumors, virtually all of them shrank, and some have even disappeared. And when we looked at what we call progression-free survival, where the tumors progressing after we started therapy, those with the low mutational burdens all progressed with a median progression-free survival of 2.3 months. And the majority of patients with mismatch repair deficiency or a high mutational burden did not progress. The overall survival showed a very similar thing, that most of the patients with mismatch repair deficiency survived, whereas by 6 months, most of the patients with mismatch repair proficient tumors, the low mutational burden, progressed. And I can tell you, unfortunately, most of these patients have passed away, whereas more than half, almost 60% of the patients with mismatch repair deficiency are still alive today. So that was the colon cancer group. We compared those who were proficient and deficient. The deficient ones with a lot of mutations did much better. But what about that other bucket? The ones for all the other tumor types. So we began looking across other tumor types, and not to our surprise, but we were very excited about this, that we could find mismatch repair deficiency in virtually any tumor type we looked at, with the predominance being in colorectal, endometrial, gastric, cholangio, and pancreatic cancer. So maybe this would work not just for colon cancer. And when we looked across all those different tumor types collected, we saw a similar sort of figure. Patients were surviving when treated with anti-PD-1. And in fact, most patients, and this is after 3 years of treatment, survived. More than half the patients survived, despite having metastatic disease at all lines of therapy, with 2 exceptions: pancreatic cancer and brain tumors. And we don't know why. Every other tumor type with this genetic signature that leads to thousands of mutations did very well, with checkpoint inhibition, so the immunotherapy, but those with pancreatic cancer and gliomas did not. So based on this work, in 2017, there was a historic approval by the FDA that approved pembrolizumab for treatment of adults and pediatrics with previously treated mismatch repair deficient tumors, regardless of tumor type. So it was tumor agnostic. That's the first time that had ever been done by the FDA. Normally, it's this drug for this disease. This time, the FDA said, we don't care what tumor it is. If they have this genetic signature, we're approving this drug. Nivolumab was then approved the next year for colorectal cancer that were mismatch repair deficient. And then in 2020, it was approved for first line, not once you failed all treatments, but initially, if you were diagnosed with mismatch repair deficiency in colon cancer, you could be treated with this agent. So that took us from the patients with advanced disease. So next, we wanted to turn our attention to patients with earlier stage disease. Remember, metastatic disease comprises about 1/3 of all tumor types. What about the early tumor types? And I have this graphic here because it highlights something I'm going to talk about, and that really I started my career working on, which is liquid biopsies and circulating tumor DNA. And so we talked about all the possible uses of these cancer genetic information, predictive markers, prognostic markers. I touched upon our work on immune antigens, but what are liquid biopsies, and how does that play a role here? Well, I'll tell you in a second. It's really marrying liquid biopsies to immunotherapy in a unique way. So liquid biopsies can really reflect anything that is in a bodily fluid, but as of the past 10 to 15 years, liquid biopsies have really gone to our understanding as short fragments of DNA that are emitted by our cells and are found in the circulation. These short fragments of DNA have a half-life that's only about 2 hours, so they're released by any cell undergoing cellular division in our body. So when you draw the blood, you can actually get a snapshot of the mutational profile or the DNA profile in the cells in your body. And the circulating tumor DNA that comes from a tumor is not very abundant, so you have to develop very specific techniques to detect that, and it's highly specific to a tumor. And you can use these for a variety of different things, mutations as biomarkers to monitor a tumor, if it's growing or if it's shrinking or responding to therapy. You can use it to track resistance. You draw a patient's blood, and are they developing genetic resistance to a therapy? You can use it to detect minimal residual disease, which is what we're going to talk about, and we'll talk about in a second. You can look at it to develop molecular remission in the same way that we do it for HCV or HIV, looking for the remission of the virus. And then something that I'm not going to talk about today, but is a very hot topic and something that we pioneered when I was at Hopkins, was that you could use these fragments for early detection of cancer. But what I'm going to talk about is using these fragments of DNA in the circulation for minimal residual disease. So what is minimal residual disease? And in solid tumors, we're a little bit far behind, because in liquid tumors, minimal residual disease is something that's commonly used, especially for things like multiple myeloma or certain leukemias. But in solid tumors and in adults, we don't have a good approach to define this. And so what is it? Well, remember I said 1/3 of cases of cancer, probably 1/4, are metastatic. The majority are early stage. And what does that mean? That means that the patient typically undergoes surgery to remove the tumor and may get chemotherapy, radiation, or in some cases immunotherapy. And no one has any idea if the patient's cured or not. The surgeon doesn't, the pathologist doesn't, the radiologist doesn't, the medical oncologist doesn't. No one. And if they say you're cured, they're lying because they have no idea. So what's the only way we currently know whether you're cured or not? Time. You have to wait 3 years, 5 years, and in some cases 10 years to know that that tumor is not coming back. So the question could we use this technology to look at mutations that were present in the tumor, to draw the patient's blood after a surgical procedure and define whether or not the patient was cured or not? And this idea really began in 2004, 2005. This was published in 2007. This is a graduate student at the time, Carla Rago, that worked with me. And we were just starting to study these fragments of DNA. And could they monitor a response to therapy? And so what we did was we put human tumors in nude mice so they wouldn't reject the tumors. And we drew from the tail vein small drops of blood, and we could, lo and behold, detect the fragments of DNA from that human tumor by looking at a human repetitive element sequencing there for a human-specific DNA fragment. And if the tumor grew, it went up, and if the tumor was treated with a therapy, it went down. But what happened if we cut it out? So when we cut it out, the cell-free DNA from that tumor disappeared. And that gave us the idea, well, maybe we should try this in humans. And so this is a human example that we published in 2008, where we were looking at mutations that we found in the tumor. This is a patient that underwent curative resection. So we took the tumor, sequenced it, found the mutations, and we had collected blood at day 0 before the surgery, day 1 after surgery, so 24 hours after surgery, day 42 after surgery, and at day 244. So day 0, before the surgery, these are mutant fragments in a gene called APC. We developed a specific probe for those mutations, drew the patient's blood, and we could find that 13.4% of the DNA in that patient's blood, cell-free DNA. So we spin the cells down, we just look at the cells in the plasma there, or the DNA in the plasma there, and we found that 13% of the plasma was from the tumor. 1 day after surgery, the red is almost completely gone. It goes from 52,000 fragments to 67. At day 42, the patient goes to see the oncologist, CT10's negative, but we see the fragments go from 67 to 326. And at day 244, the CT scan's positive for recurrence. At that point, we knew the patient recurred. And there was almost 2,500 fragments of that APC mutation in the blood. We knew at day one that patient was going to recur. Well, you could say, well, isn't that just residual DNA fragments from the resection? By 24 hours, knowing that the half-life was every 2 hours, and let's say we give it 5 half-lives just to be generous, by that time it should be completely gone. And we still saw fragments there. So we knew at day one it was still there. At day 42, we also knew it was there. So maybe this technology could be used to detect minimal residual disease after a curative resection to help inform us that there's a high likelihood the patient's cured or not. And when we looked at a number of patients back in the study undergoing curative resection, and we drew the blood between 6 to 8 weeks after that curative resection, those patients with undetectable levels went on to be disease-free. Patients with detectable levels all recurred. And that's what each of these steps are. It's a recurrence. Patients recurred that were positive for circulating tumor DNA. What brings us to this marriage between liquid biopsies, immunotherapy, and cancer genetics. With Yelena Janjigian, when I moved from Johns Hopkins to Memorial Sloan Kettering, we decided to try this project. And this were patients with mismatched deficient tumors, lots of mutations, that were undergoing curative resection. The patients underwent what's called an R0 resection. That means they got all the tumor out, visible tumor. We sequenced the tumor, confirmed that was mismatched deficient, and then used those mutations to design an assay to look in the blood to see if there's any tumor left. And if the patient after surgery, after adjuvant therapy, had no circulating tumor DNA that we could detect in the blood, we just sent them for observation. If they had circulating tumor DNA in the blood, we randomized them to anti-PD-1 treatment or placebo. And so we screened 174 patients undergoing curative resection, and we found 125 were negative for circulating tumor DNA and 25 were positive. And those 125 that were negative, by and large, over the course of what we've been following at this point, have been negative. We were wrong 7% of the time, 7 times out of 125, so a little less than 7%. Those that were positive, virtually all of them recurred to date. And so they've recurred over the course of our study here. They did not receive anti-PD-1. But what about the ones that were positive and received anti-PD-1? Will they look more like the red line or will they look more like the blue line? And so when we looked at these patients, they looked as if they were circulating tumor DNA negative. We knew from previous history and dozens of papers that if you're circulating tumor DNA positive, you're going to recur with a positive predictive value in the 90%. So changing the natural history of that positivity because we could detect the patients that were negative. So we thought that that was pretty exciting, and we're still following these patients. And my hope is that this will be reported relatively soon. So what about early stage cancer? And this is a patient that I'll talk about at the end that we wanted to look at. So we went from metastatic patients to adjuvant post-surgical patients. What if we move this to before surgery? And so we decided to focus on surgeries that were particularly brutal and look for patients that were mismatched repair deficient, and rectal cancer fit the bill. About 5 to 10% of rectal cancer are mismatched repair deficient. There's about 60,000 patients worldwide with mismatched repair deficient rectal cancer. They're resistant to chemotherapy. We knew checkpoint blockade was very effective in the metastatic setting and in the adjuvant setting. And we knew that most of the surgery here was a combination of chemotherapy, radiation, and surgery, where they remove the rectum, and it has life-altering consequences. It leads to infertility because of the chemotherapy and radiation used, and because of the surgery subsequently. And more than 30% of patients have an ostomy bag for the rest of their life. So with Andreas Sursek, we decided to do this study in these patients with rectal cancer, early stage, not metastatic, who would normally get chemotherapy, radiation, and surgical resection. And we found these patients that had mismatched repair deficiencies, so lots of mutations. And we treated them with 6 months with a different anti-PD-1 drug called dostarlimab. And if at the end of 6 months there was any residual tumor, because we didn't want to lose opportunity to cure these patients, they underwent chemoradiation and surgery. But if by chance the tumor was gone, and this was really Andrea's decision here to incorporate this in study, we decided to follow them up non-operatively. We would not do chemotherapy, would not do radiation, we would not do surgery. And so we did our first patient, and you may not be accustomed to seeing a tumor up front and live, but that's an endoscopic version of a rectal tumor. Very large, very friable. Patient's very symptomatic here. Here's an MRI of the rectum, and the arrow shows where the tumor is, and a PET CT scan of the rectal tumor as well. After 6 weeks of therapy, and we were watching these patients very carefully, we saw a dramatic change in the tumor. And at 3 months, we saw a complete eradication of the tumor. And that was also confirmed by MRI and PET scan. And we continued to follow this patient. For 22 months, the tumor remained gone, despite only 6 months of therapy. So our first patient was a success. 38-year-old, very large tumor, very symptomatic, and had a complete response by a physical exam, endoscopic exam, and by MRI. So then we did the next 14 patients, and we had the same result. Every single patient had elimination or eradication of that tumor. And that was a huge surprise to us. And when we followed these patients over time, those tumors remained gone. We eradicated them and they remained gone. And we have now moved from those initial 14 patients that we reported. We're now up to 42 patients treated. And we actually have an 100-patient cohort that we're going to report on as well. And every single patient remains a complete response and disease-free to date. And really our cutoff here was, could they remain disease-free for 12 months? And they've remained disease-free for much longer. So I've taken you down this path of using this initial hypothesis from metastatic setting to adjuvant setting to preoperative setting. What about prevention? Because that's really what we want to do. And talk to you a little bit about some work we've done here, and then move slightly in a different direction towards the end of the talk. So Lynch syndrome, remember the hereditary form that's 10 to 15% of mismatch or deficient? See, there's about 1.5 people in the United States alone living with Lynch syndrome. They have a rate of 4% per year get a cancer. And they can get cancers anywhere, small intestine typically, or large intestine colon cancer, rectum, endometrium. But really, they get them anywhere. And there's germline mutations and risks associated to which germline mutation they have that leads to that mismatch or deficiency. So with Sophia Sadler and Emily Harold, we had a question, which was, could PD-1 blockade that works so well in the metastatic patients and the adjuvant patients and the patients with rectal cancer work in the prevention of the precursors of colon cancer, the polyps? Could they prevent the development of polyps in these patients with Lynch syndrome? That was the one question. We thought it was a good hypothesis and may work. And number two, could PD-1 blockade prevent or delay tumor formation in these patients, since we knew they're developing tumors at a rate of 4% per year? So we have a registry of 837 of these patients that we follow. And we could do an observational study and say, okay, we know which patients received checkpoint inhibition and which ones didn't. And could we look at these questions in that regard? And so the first question was, could we prevent the development of these polyps? And we looked at patients who received immunotherapy and had not received immunotherapy, and there was no difference, which was shocking to us. We thought we would definitely see a difference in the number of polyps that we could form. What about the number of polyps for colonoscopy? This is the baseline incidence of polyps. This is the number of polyps, and there was no difference whether you received immune checkpoint blockade or not. So what about the second question? Could they prevent cancer in these high-risk Lynch patients treated with immune checkpoints? And this is the incidence of cancer in these patients that did not receive immune checkpoint. But if they did, we could decrease the incidence of cancer development in these patients by about 80%. So a little bit of a conundrum. We can't prevent the polyps that eventually turn into cancer, but we can prevent the cancer. And this is just another way to look at this. The 5-year the second cancer incidence over 5 years per patient in patients who did not receive checkpoint blockade versus those. One interesting caveat. If the patient had received chemotherapy, not immunotherapy, we also saw a decrease in the amount of cancers formed by these tumors. So opening up an idea that immunoprevention may be a real thing. So what does this tell us about immunity in Lynch syndrome and in mismatch repair? We know that the normal tissue is typically non-immunogenic. It's what we call mismatch repair proficient, and it's microsatellite stable. And the tumor mutational burden, or the amount of mutations, is low. In pre-malignant or in polyps, we also know they're non-immunogenic. We know if they receive immune checkpoint blockades, they still develop. We also know that most of them are mismatch repair proficient, and most are microsatellite stable. So they don't have that thing called microsatellite instability. And they have a tumor mutational burden that's still relatively low. We also know that the patients who are locally invasive, like the rectal cancer patients I showed you, are mismatch repair deficient. They're microsatellite unstable, and they have lots of mutations. And then eventually, once you get to the metastatic setting, we're not curing 100% of them like we are with the early stage rectal cancers. So they're less immunogenic, but we still know they're mismatch repair deficient, microsatellite unstable, and they have a high mutational burden. So giving us some insight into the immunobiology of these tumors and how that relates to the genomics and to what we see clinically. So in summary about prevention, checkpoint blockade does not inhibit polyp formation. It does slow tumor development. And so this data suggests that immunogenicity develops over time, and there's a window of opportunity during oncogenesis of these mismatch repair deficient tumors where they're non-immunogenic, and thereafter we can treat them with immunotherapy. So I talked to you about the clinical features. I'm going to dive in a little bit about the molecular features of these tumors. And then I'm going to end with an approach based on what we've learned, where we're trying to convert mismatch proficient tumors into ones that are deficient tumors. So what do we know? So in terms of tumor mutational burden, we know that these mismatch proficient tumors have a lot of mutations. And two folks in my lab, Benoit Rousseau and Mike Foote, started asking the question, is it just because they have a high mutational burden? And actually, there's even an FDA approval saying that if you have more than 10 mutations per megabase, you can get treated with anti-PD-1. But we wanted to know how much that was due to the type of mutation or the condition that you have, or was it just because you have a lot of mutations? And so what Mike and Benoit did was the following. They took tumors with a high mutational burden here and stripped out those that were mismatch repair deficient or had another mutator phenotype called polymerase D deficient, and looked at the tumors that had a high mutational burden that didn't have these molecular signatures. And what we found that is despite them having a high mutational burden, they were not immunogenic in the same way that patients were with low mutational burdens. So there was something special about these genetic signatures that made them more immunogenic. But when we looked a little bit more closely, what we found was that there were some tumors that were tumor mutational burden sensitive, whereas there were other tumors that were tumor mutational burden insensitive. And when we looked at the tumors that were tumor mutational burden sensitive, there were things like non-small cell lung cancer, melanoma, and head and neck cancer. And I want you to say this in your back of your heads for the end of the talk. These are all tumors that were caused by environmental mitogens. Melanoma, UV light, non-small cell lung cancer, head and neck cancer, tobacco smoke, or tobacco itself. Whereas these other tumors may be caused by other etiologies, but they certainly haven't been linked to exogenous environmental factors. So that gave us the idea, okay, so if it's not just a lot of mutations, could it be mutation class? And in our work on liquid biopsies, we knew that we could detect mutations by the degree of foreignness, because we could create probes that could detect mutations that looked more foreign to the self versus those that were not very foreign. And we decided to borrow that and use that for our immune antigen discussion. So we knew things like gene applications, large deletions, point mutations didn't look very foreign. They actually looked a lot like yourself. but things like gene rearrangements, insertion and deletions, and frame shift mutations, which are just insertion and deletions that are not divisible by 3 that really change the protein, probably are the most foreign. So we did a very simple experiment. We started looking at individual genes, and if they were altered, we did what's called a blast search, but in reverse. We wanted to get a score on how different a mutation made an individual protein if it was expressed as a protein. And then we took a collection of mutations for a given tumor, and that gave us how different those mutations would make that tumor if they were all expressed. And that gave us what we called an alien peptide score. How foreign or how different or how unique were the collection of mutations if they were expressed as protein from the rest of your self-peptidome. And so when we did this and we looked across every tumor type that was in this database called the TCGA, the tumors with the most foreign appearing peptides were these mismatch repetion tumors, followed by the polymerase E and polymerase D mutations, which is another syndrome that causes hypermutability, and then non-small cell lung cancer and renal cancer. So giving us some credence to the thought that maybe the foreignness of the mutation had a big impact. So what type of mutations were the most foreign? Well, if you looked at all the mutation categories and we separated them down to 9 mers, because that's how the immune system oftentimes see these in their MHC class 1, which is how they're presented to the immune system, the most foreign-looking peptides were these frame shift mutations or in-frame mutations and sometimes missense splicing mutations. So giving us a clue that these frame shift mutations were maybe the most immunogenic. What about the evolution of immunogenicity? And I touched upon this a little bit in the prevention area. We did these experiments a number of years ago with Tim Chan when he was still at Memorial, where we knocked out mismatch repair deficiency in these cell lines in mice. And we wanted to see two things. What is the link between immunogenicity and the genomics? And how did this happen over time? What were the kinetics of this evolution? So one thing I can tell you right now is that if you knock out mismatch repair deficiency, the next day the tumor is not immunogenic. Also, the tumor doesn't have a high mutational burden. So you have to wait weeks and weeks and months before that tumor cell becomes immunogenic. It has to accumulate mutations over time. And sometimes over a year, you have to passage these cells. They'll acquire these mutations, and then they'll become immunogenic. And what we found here was in 2 different cell lines where we could knock out mismatch deficiency, that these cell lines became immunogenic over time, and that this was proportional to the amount of mutations they accumulated over time. So what had we learned about just simply about these mismatch repair tumors? They had a loss of mismatch repair. They had a lot of mutations. These mutations were enriched for insertion deletions, but specifically frame shift mutations. I'm not going to touch about this, but they were highly diverse and they were highly subclonal. And we knew immunogenicity doesn't happen overnight. It takes months to years. So we had a question. Could we pharmacologically recapitulate mismatch repair deficiency in tumors that were mismatch repair proficient? What we do know is that mismatch deficiency only affects about 5% of all cancers. The remainder are mismatch repair proficient. So could we pharmacologically recapitulate mismatch repair deficiency? And so this is the last part I'll talk about, which is, can we chemically hack our genome to induce an immunoresponsive phenotype? And so what's the current paradigm? And I'm going to say it's passive. We have a tumor and we have a target. You have to, by accident, find the target. If you're lucky, you have mismatch repair deficiency, or if you're lucky, you have a BCR-ABL mutation, or if you're lucky, you have HER2 amplification. Those are all treatable therapies. And then you get a therapeutic response. But we want to do the opposite. We wanted to hack the target into the tumor and then give the appropriate therapy and induce a therapeutic response. So our hypothesis was, could we chemically alter DNA for the desired genotype to lead to immunogenicity? And the barriers primarily that were told to us by people outside our group were you're going to induce random mutagenesis. You'll have a subclonal architecture, and you need clonal neoantigens for them to be immunogenic. So it's not going to work. And the second was we're going to give mutagens to alter the DNA. You're going to result in more cancers. And so the rationale was that we knew that these tumors that were mismatch repair deficient were exquisitely sensitive to immunotherapy compared to their microsatellite stable or mismatch repair protein counterparts. And we knew that we needed to enrich for single base pair substitutions and indels, and we knew that we needed a high mutational burden, specifically in frame shifts. So could we recapitulate this? And so I'm going to dive into the example that actually allowed us to do this. And it was a combination of two drugs, one called temozolomide and the second called cisplatin. They're both used clinically today. But temozolomide did one thing. It induces a lot of mutations when you give it. Tumors that are treated with that, if you sequence that tumor after treatment, it induces a lot of mutations. And cisplatin induces insertion deletions. And we looked at a number of other agents, and I'm going to draw your attention to DMBA and ditronol. DMBA is a drug that also does the same thing. It induces a lot of mutations and a lot of frame shifts, and it's a component of tobacco smoke. And ditronol also induces a lot of mutations, a lot of frame shifts, and that's a component of TAR. So these are agents that are probably cancer-causing, especially in those tumors that are high mutational burden sensitive that I described earlier today. But what I'm going to focus on are these two other agents, temozolomide and cisplatin, that induce a high mutational burden and induce a lot of indels to show you what we found. So Benoit Rousseau in our laboratory did these experiments. He was the only one crazy enough to take on this experiment, which was, can we mutagenize tumors to make them immunogenic? So we took tumors that were non-immunogenic from mouse lines, treated them with these immunogenic drugs over 8 weeks, washed it out, and then looked at the genetic landscape, and then looked at immunogenicity. And when we looked at the genomic landscape after treatment for 8 weeks, we saw a number of things. No drugs, almost no induction of mutations, no induction of frame shifts, and they remained microsatellite stable. We really didn't see a change in their mismatch repair deficiency profile. If you gave cisplatin, you saw a few more mutations. You saw what looked on the graph like a clonal peak, but the mutational burden didn't go up that high. If you give temozolomide, the number of mutations per megabase went up significantly, especially in missense mutations. But if you combine cisplatin with temozolomide, the number of mutations went up tremendously, especially frame shift mutations. And when we looked across these tumor types, we could see that temozolomide and cisplatin induced not only missense mutations, but frame shift mutations, a gain in mutational burden, and that made these tumors microsatellite stable, just like the mismatch repair deficient tumors. When we looked at the mutational signature of these tumors, they were largely in the combination consistent with mismatch repair deficiency. And when we put them in immunodeficient mice, whether they were treated with vehicle or temozolomide alone or cisplatin alone, or temozolomide plus cisplatin, or the control knockout cell line, they all grew the same in immunodeficient mice. But if you put them into mice with immune system, the vehicle, the temozolomide alone, the cisplatin alone, all grew normally. But the ones that were treated with a combination grew as slowly as our control cell lines that were mismatch repair deficient. And then when we put them into mice and treated them either with control antibody or anti-PD-1, I can tell you the combination of temozolomide and cisplatin that had many, many mutations, many, many frame shifts. When you treated them, not only did they have a delayed growth, but if you treat with anti-PD-1, you eradicated them. Whereas if you treat them with an isotype control antibody, you just slowed the growth. And this was even more so than with the MSH2 positive control knockout. All the other cell lines grew as if they hadn't seen an anti-PD-1 agent and they weren't delayed in their growth. When we looked at immunoediting, so the resulting tumors, their immunoprofile, those that were treated with a combination, when we did force tumors to be grown in these contexts, we saw immunodeletion or a heavy immunoediting in missense mutations, heavy immunoediting and frame shift mutations, almost exclusively to the combination group. And obviously a huge amount of TMB loss in the combination group as well. And when not only we looked at the molecular signature in these tumors, that the combination showed a high enrichment for mismatch deficiency there in the red, but when we looked at the tumor themselves, those with the combination, and it almost looks fake. You can see a very pale expression of these mismatch repair proteins, MSH2 and MSH6. And the loss of those, so that not only did they become genetically like mismatch repair, they actually lost their mismatch repair deficient proteins. So what we saw was that these tumors that were treated with temozolomide and cisplatin for 8 weeks, they looked like they had a higher immunosuppressant sensitivity, and I didn't show the data through class 1 presentation. They had preferential immune editing related to mutations. They had persistent mismatch repair deficient inhibition, and they were very sensitive to checkpoint blockade with anti-PD-1 in mice. And then when we did this experiment, we took mice with mismatch repair proficient tumors, treated with either vehicle control or temozolomide cisplatin, and treated with either anti-PD-1 or a control antibody. Those that were treated with anti-PD-1 lived longer, presumably because we induced mutations that then could respond to anti-PD-1, whereas if they were treated with IgG, they continued to grow. So these mice here that were treated with vehicle all died. Patients that were, or the mice that were treated with temozolomide and cisplatin had a little bit of delayed in growth, but they still all died. Whereas those that were treated with anti-PD-1 that had been treated with temozolomide and cisplatin, you had some mice that were cured. One question to us, would there be an effect on healthy tissue? And we saw a minimal effect on healthy tissue. The mutations for megabase were very low, even with the combination with PD-1 blockade. So then we decided to take this to clinical trial. We had seen enough data to make us enthusiastic that this just might work with patients. And so we treated patients with advanced disease with temozolomide, cisplatin, and an anti-PD-1 agent called nivolumab. And what we saw when we took the whole group together is that there was an absolute increase in the amount of mutations that we were inducing across these tumor types. And when we looked in the circulation of these patients with circulating tumor DNA, we saw accumulation of molecular signatures that were consistent with mismatch repair. But I'll leave you with this. And the patients that developed both a mutational signature consistent with microsatellite instability or mismatch repair deficiency and accumulate a lot of mutations, they did significantly better than the patients that did not. So giving us a hint that we could change the outcome of these patients by hacking their genomes with these chemicals to induce an environment that was more immunogenic. So in conclusion from the clinical trials, we think that temozolomide and cisplatin induces a mismatch repair deficient genotype and phenotype through the inhibition of mismatch of microcellulite or MSH2, which is a microcellulite or a mismatch repair deficiency repair protein, by increasing the sensitivity to PD-1 blockade. We think these combinations spares healthy tissue because we think that cancer cells are more susceptible to DNA damage. The clinical trial with temozolomide, cisplatin, and anti-PD-1 improved overall survival if the patient acquired a mismatch repair deficient genotype along with a high TMB. And we believe that these results warrant further investigation. We're actively investigating these now because we think that we'll give patients without mismatch repair deficiency a chance to really to benefit from checkpoint blockade in the same way our patients have. So overall, I think this is an overall example of how cancer genomics has really aided the management of colorectal cancer. We think tumor intrinsic genotypes like mismatch repair deficiency. I didn't touch on POLI and TMB are highly associated and predictive of exquisite sensitivity to immunotherapy or checkpoint blockade. And we believe that neoadjuvant therapy may pave the way for immunoablative therapy that may replace the need for surgery, chemotherapy, and radiation for many tumors that harbor these genotypes. So obviously, I want to thank many of the giants that preceded me, not only my mentor, Bert Volosine, but also Henry Lynch that described Lynch syndrome and developed the theory for genetically based cancers, and Albert de la Chapelle, who listed the genetics of hereditary colon cancer. Obviously, the patients in the family and the many funders of this work, all the people that contributed to this research, which has been tremendous. and our lab at Memorial Sloan Kettering. This is in Central Park. And thank you very much, and happy to take any questions.
A great insight into where the power of molecular genetics, immunology, and the modern science is allowing really great progress in cancer. So very exciting and inspiring. Room for questions. We could have questions from the room. Okay, I'll start back there. Jamie? And you should be able to be picked up on the mic and speak up.
All right, so thanks very much. That's really, really encouraging and exciting to see. I guess I was interested to know about the HLA presentation machinery itself.
Yeah.
And that not getting mutated and like how does that fit into the whole equation?
That's a great question. So when you look at mismatch deficient tumors, unlike melanoma and other tumors, you actually see a high frequency of class 1 loss. You actually get biallelic mutations in a protein called B2M, which is a chaperone for HLA class 1. And that leads to loss of expression of HLA class 1. In those tumors, which is about 40 to 60% of mismatch deficient tumors, they still respond to therapy. So there are other effector arms of the immune system that are involved in this. So we've described CD8 cells, CD4 cells, gamma delta T cells, and NK cells, which are very potent contributors to this response. And there may be other mechanisms involved here as well. So even if the antigen presentation machinery is rendered or neutralized by mutations or other things, we still see a response in these patients.
Jess?
A great talk. Thank you so much. I'm just wondering if instead of inducing immunogenicity with increasing mutational burden, if you can increase immune cell infiltration to the tumors and if that would make them more sensitive to immunotherapy.
Yeah, and I think a lot of people have thought that. I think just from my genetics background, it's kind of a myopic perspective, but we believe that cancer is tumor intrinsic, and for a proper immune response, we need that tumor cell to look foreign. And most tumor cells don't look foreign. And even taking a step back, I think that there's a very early decision point where a tumor either develops what we call in our lab a couple superpowers. Either one superpower is that you become invisible, and if you're invisible, then you can acquire these mutations that are immunogenic. And if you're not invisible, then you acquire the ability to never die, of immortality. And those tumors, if you look at the quality and nature of their mutations, they're not immunogenic. They're not expressed in class 1 or not. So even if you forced cells there, it's unlikely you get an adaptive immune response. You can get an innate immune response, and the cytokines may cause some cell death, but it won't be adaptive and it won't be tumor specific, if that makes sense.
Mason?
Thank you. I was just curious what proportion of patients ultimately did develop the mismatch repair deficient genotype. And then if you think with sufficient power in the future, if you'll be able to retrospectively identify the patients that you think will ultimately develop your genotype of interest.
Yeah, I think to me, this was a failed study because I was hoping that most patients would develop the genotype. And we only saw the genotype and the high tumor mutational burden develop in like 15 to 20% of patients. And I'll give you the answer already that I think even before the paper's out, why we think it failed. Which is that as you develop these mutations in vivo, they're immunoedited immediately. So you never give the tumor cell the opportunity to actually begin to accumulate these mutations. Because as soon as you induce some immunogenic mutation, that's immunoedited out. So that cell's lost. So what remains are the cells that you didn't transform or that you didn't hack. So I think future approaches, and we're doing this now in the lab, will have to somehow cloak that cell while it's accumulating mutations. Does that make sense?
Yes, thank you.
So we have a question online. We'll get to one more in the audience, and then we're going to have to get Dr. Diaz on the way to get his pizza and back to the airport. So Danielle Samini from the Biology Department at Tech asks online, do the Lynch syndrome patients have to stay on the anti-PD-1 therapy for the rest of their life? And is there a risk of autoimmune reaction?
Yeah, no, I think that's a great question. So what we stopped therapy after 2 years. And they probably don't need 2 years of therapy. In fact, with the rectal cancer patients, we only treat them for 6 months. But rectal cancer is early stage localized tumor. We do think that the metastatic scenario is very different. And if I can paint a picture for you, I think that there is, and we know that there's a very different immune environment that's localized versus one that is within a lymph node versus one that's metastatic. And I think those immunologic niches have different requirements for duration of therapy. We know that the localized ones, let's say in the mucosa, need a shorter course than ones that involve the lymph nodes, than ones that involve distant metastases. So the longest I've seen is 2 years. We think we can probably pare that down, but no shorter than I think 4 to 6 months. In terms of autoimmune reactions, we absolutely have seen them. And if there is a pre-existing autoreactive T cell or B cell, oftentimes those do get stimulated, and the severity of that clinical syndrome can vary.
So Anthony, go ahead.
So I'm wondering if the cells with the greater mutation burden that are more obvious to the immune system, do they die differently? Are they regulated differently in terms of clearing the tumor tissue itself? Because clearly it goes away. So how is it that those cells are actually are they more susceptible to apoptotic death? How does it work?
You know, that's a great question. And I don't think we know that the mechanism of immune killing can be diverse. It can be cytotoxic T cells. We know it can be gamma delta cells or NK cells, but the cleanup and number one and number two, do they die differently? We don't know. But that's a great question.
Okay, we'll squeeze in one last quick question. Go ahead, Zin.
Great talk, and I'd like
Thank
to ask a general question. You want to make the cancer cells more foreign so you can attack them.
Right.
I have some evidence that exercise can change PD-1 expression in monocyte and some cells. My question is whether you can come up with a method to kind of make your immune system more sensitive to maybe less foreign cancer cells.
So more sensitive to yeah, that's an interesting question. So the current thinking is that checkpoint expression like PD-1 is a break. So let's say you have a really bad infection, or let's say you have really bad inflammation. What's bad? For example, a heart attack, or if you have a cut, that environment will upregulate checkpoints towards the end of that inflammation because it doesn't want the immune system to go crazy. It puts on the brakes. Cancer has co-opted that to slow things down. So your first question kind of makes me think, why would exercise induce PD-1 expression, right? And so the question is, are you inducing some low-level inflammation during the exercise? And that's inducing upregulation of immune checkpoints. The second thing is we know cancer patients that exercise do better. And so could that be a link? But then the third question, which is really interesting, is if you upregulate it during exercise, could that detect tumors that are more subtly foreign? And I think it all depends to the degrees of what the immune system that individual immune system will allow, you know. But it's a great question. It's a great observation too.
So please join me in thanking Dr. Diaz.
Thank you very much.
Please, thank you very much. Safe travels. Have a great dinner. We'll be in touch tomorrow.
Thank you. Take care. Bye-bye.